Fellowship Committee for Travel Fellowship Committee for getting me here. My name is Ben Glicksberg. I'm a PhD student at the Icahn School of Medicine at Mount Sinai in the labs of Dr. Joel Dudley and Rong Chen. And like the previous talks, I'm going to also be talking about electronic medical records, the racial disparities in disease networks. So we can study disease in many ways, as we've seen in this conference so far, but for the current talk, we're going to be talking about it in the context of epidemiology, where in the most basic sense, a disease is at a nexus between genetic architecture and the environment in which someone is raised. Furthermore, our idea of race, or the construct of race, is also the product of one's genetic background as well as the environment in which they're growing up. And so this is studying disease in the context of race is important and interesting for research purposes, but also for clinical purposes, where a disease susceptibility can be different for people of different races, of different backgrounds, but also progression of disease into other comorbid conditions can differ based on race, which is very useful to know because it can tailor clinical management, personalized interventional therapies, and may warrant follow-up investigation on why these diseases progress in this way. So in the field of disease networks, there's been many pioneering and interesting works, and this isn't an extensive list, this is just a few of them. Go et al. Built a human disease network and a human phenotype network connecting diseases based on shared genetic architecture. Hidalgo et al. Furthered this work by actually including clinical data in the form of Medicare data in ICD-9 codes for elderly patients 65 and older. And actually, another great recent study was by Jensen et al., where they took the entire population of Denmark, covering all 6.2 million people, and extended the idea of comorbidities beyond simple connections to look at the trajectories of disease and how they might differ, as we saw in the last talk. Just to note, the senior author, Dr. Brunak, is going to give a keynote later, and it'll be interesting to see how he describes this study. While these studies are really interesting, there are some gaps that still need to be filled. And this isn't a flaw of the studies, it's just based on the data availability. The previous studies I mentioned and many others are mainly constructed using individuals of European background. And furthermore, the differences between the races aren't the focal points of these studies. And the reason this is important is especially because Hispanic Latino populations are critically understudied in healthcare analytics. This is important because many studies reveal many different disparities in outcomes between racial groups. And this could be the case because physicians often have to bridge the inferential gap where they treat a patient of a certain race using research and knowledge that was from studies studying a different individual of racial background. So with this in mind, we first wanted to look at how does racial background affect disease susceptibility, and furthermore, disease trajectories differ between these racial populations. So for our study, we used data from Mount Sinai Hospital, which is located in New York City. Mount Sinai stores its data in the Mount Sinai Data Warehouse, where there's over 4 million patients and 16 million patient visits. The Mount Sinai Data Warehouse de-identifies these data into electronic medical records, where investigators can apply for IRB access and get access to these. As we've seen before, EMR data can have disease diagnoses, demographic data, and drug information, among many other things. One thing that's amazing about New York City, this is census data from 2010, we can see that there is a lot of heterogeneity in its population. Even though there's some clustering of European American Caucasians in Midtown, Mount Sinai Hospital is actually located at this border between Upper East Side and East Harlem, which houses a lot more racial diversity. And because of this, the Mount Sinai Hospital has a pretty decent representation of underrepresented minorities of over 4 million people. further refine this data, we only included individuals that had a hospital visit since 2003 when the EMR was implemented at Mount Sinai. And we further only kept people that had a reported sex, age, and race, no individuals that with unknown or unreported. And finally, we studied only the 3 populations with the most representation in our cohort, which was EA, Caucasian, African American, and Hispanic Latino. So after these steps, we're left with over 1 million people and a pretty decent representation of Hispanic, Latino, and African Americans at 17% and 22% respectively. So in the current study, we have to first deal with the EMR data. As we've seen, it's not optimized for research purposes, it's really optimized for billing. So we have to pre-process the data and classify them and organize it. Then we can determine disease susceptibility profiles that may differ based on these races. And we could follow that up with actually going beyond simple disease susceptibilities to how diseases are connected to one another and if they have some temporal ordering. Finally, we can extend these connections to build disease-specific networks and do some interpretation on them. So as I mentioned, EMRs and especially ICD-9 codes are not meant for research purposes. They were really built around billing. And because of this, ICD codes are organized in a tree structure, but that aren't necessarily together in some meaningful disease relationship. So luckily there is such ontology, such as disease ontology, which connect these disparate codes to a disease term, which is more precise than a general category of diseases. So out of the 2,000 disease terms in disease ontology that have at least one ICD code, we can further refine them to diseases that have at least 10 people of each racial background in our cohort, which leaves about 1,200 diseases. Here we can see just a frequency plot. There are many diseases in which there are the minimum representation of 30 people, but there are also some very, very high diseases of very high prevalence, and these turn out to be hypertension and diabetes-related diseases. We can further classify these diseases into broader groups, and there's a great software called the Clinical Classification Software by the HCP organization that categorizes the diseases in many ways. We use the single-level categorization, which has 202 categories, and this could be intestinal infections, for example. And we further only kept codes that have categories that have at least 5 ICD-9 codes encompassed in them. And we can see here in a radial plot the proportion of, or the amount of categories and how many ICD codes they have. And we see some of them have the minimum 5, but we do see some categories with large amounts of diseases. And these tend to be some of the more broader diseases, such as cancers. So with these data organized and processed, we can begin to do some analyses. The first question we can ask is, how does disease susceptibility differ between racial groups? And to answer that, we did a simple logistic regression where we asked, does the probability of a disease depend on self-reported race, sex, and age? And age here is their current age. To assess the differences, we looked at the significance and beta size of the race variable. And just to point out here, when you're doing logistic regression, you need to compare the variable to a baseline. So the baseline in this case was the people of European ancestry. So any difference is going to be, does African American or Hispanic Latino individuals differ to Caucasian individuals? So out of the 1,200 diseases that we started with, we found 81% of them, a large percent of them, have some difference in susceptibility between the races. Here we can show the distribution of Bonferroni's corrected significant associations. On the left side, we see African Americans, it's cut off, and on the right, we see Hispanic Latino. And the beta values on the upper side correspond to increased risk as compared to European Americans, and below 0 is a decreased risk. So some of these diseases that are highlighted from a clinician in our group make clinical sense. Sickle cell anemia floats to the top of the African American risk, And a lot of skin disorders, individuals of African American and Hispanic Latino are protective. There's increased risk for European Americans. We can also notice a trend here where for the African Americans, it seems like the majority of diseases are at an increased risk compared to Caucasians. And for Hispanic Latinos, it's the opposite trend, which we can keep in mind for the rest of the talk. So we can now go beyond simple disease susceptibility to see whether two diseases are connected, and if they're connected, are they temporally ordered, and does this ordering differ between races? So we took all 1,200 diseases, we took all unique combination of disease pairs, we then only kept pairs of diseases in which there were at least 10 people of each race that had both conditions. So after that filtering step, we were left with 37,000 diseases. Then we went into the records of these people, and we said, in their timeline of visits, we took the earliest indication of that disease, and we counted simply how many people have disease A before disease B, as we can see in this depiction here, or disease B before disease A, or they could be coming up at the same time. And this could be in actually organic, where two diseases are manifesting in close proximity, or it can be an artifact where someone is coming into the hospital later after presenting with both diseases, and that could be they've had it for years, and this is just a limitation of the data. So if we found that one disease significantly preceded another more times, we did a cumulative binomial probability where we gave each disease a 50% chance to occur before the other. And we end up finding that 2,000 diseases for EA are temporally ordered, over 3,000 for AA, and only 690 for Hispanic Latino, which is another interesting thing to keep in mind, that many of the diseases were not temporally ordered. And this may be a reflection of other things that we'll talk about later. So just because two diseases are ordered temporally doesn't mean that they're actually connected if they're significantly comorbid. And the way we assess that is we did another logistic regression where we took the pairs that were had significant ordering, and we said, does the probability of the second disease depend on having knowledge of the first one, of presenting with the first one? So we looked at the beta and the significance of this term when controlling for age and sex. And now we can see the distribution. These are the distribution of significant disease pairs between each population. And the first thing we notice, again, is that the African Americans have the largest proportion of disease pairs that are significant. And we can also look at the categorization of the diseases that are the response diseases, that are the diseases that are the sequelae, the ones that are being led to, and compare them across all populations. One thing that's interesting is that the Hispanic, Latino, and African American populations are enriched for a lot of ocular and vision-related diseases such as cataract, glaucoma, and other retinal detachments. What's also interesting is the only category that was enriched for the disease pairs common among all three were mycoses, which may make sense as people in New York City live very close together, and fungal infections could be a product of environment. So it could make sense that maybe there is a strong environmental component to this that extends beyond race. So with these pairs, we can actually build race-specific networks, and this is a very abstracted overview representation of them. They're actually made up of connected nodes that are ordered and have magnitude. And I'm getting a little out of time, so I'm not going to go compare them directly, but we do notice that topological differences may reveal some biological underpinnings of these disease pairs. Furthermore, we can move beyond the networks themselves to look at other factors relating to this networks, and one of them is whether how the hub diseases, which we define as diseases that have 10 or more outward directed edges, how do they differ between the populations? And the first thing that's apparent is that there is some stratification where there are diseases that are very prominent and leading to many other diseases that are unique to each population. Well, specifically for the African American and European American populations, what's interesting to note, first off, is that there are no hubs that are unique to the Hispanic Latino population, which goes along with the trend that we saw from the previous findings. So there were 27 hubs for the Caucasian group, a whopping 44 for the African American. And because the previous study used more higher level categorical representations to the disease, we can actually compare our results to the Hidalgo et al. Study and to the Jensen et al. Study to see, do our results coincide with them? Does it correlate? And it does for many of these diseases. Hidalgo et al. Found that aortic valve disease and mitral valve disease were much more comorbid in European Americans and African Americans, which we found as well. They also found that asthma-related disorders in the African American population were much more comorbid than European Americans, which we found as well. Furthermore, they found that type 2 diabetes and hypertension were significantly comorbid in both populations, but were more so in African Americans. And we actually found that as well. Although these diseases, it's hard to see on the screen, although they are hubs of all three populations, they were significantly more enriched in African Americans, having the most connections between them. Furthermore, this analysis the disparity in these hubs may reflect some clinical need. There are certain diseases in the African American only hubs that may stem from these diseases not being managed properly. And some of them are known, like hepatitis C and type 1 diabetes, but even diseases like constipation and diaper rash could just not be addressed properly in the population, which could direct future research for clinical care. And lastly, I just want to say that just because a disease was not a hub for a population doesn't mean that it's not interesting. These are the top 5 connections in the Hispanic Latino network. And one thing that's really interesting is that 3 out of the top 5 have connections related to some mental disorder or mental disease, which is also noted in the literature that despite lower levels of prevalence, that it's still a problem that goes unaddressed. So with these findings, we have a lot of future actions that we're already in the process of doing. First off, Mount Sinai just joined a Health Partners Consortium where they have centers in different boroughs. So we're going to get a broader scope of data, new hospital data, and we're also going to control for socioeconomic factors within the data that we already have. I also mentioned that we looked at order of disease, but we didn't really look at the latency between the diseases. We calculated the average latency for each race, but we didn't really delve into it and understand why. We also just got access to environmental exposure, such as allergens and pollution, that we're going to include into our model. And lastly, I just want to make a very important point that even though we define these races based on self-reported race, there's actually a huge amount of genetic ancestry heterogeneity within it and population substructure, which may confound and mask the results. Mount Sinai has a biobank of 10,000 people that we have genetic data for. And I did a PCA analysis where I'm separating the population between the first and second PC, and I've overlaid the 1,000 genome reference panel. So we can see that, it's very hard to see, but European Americans are at the top, Han Chinese are on the bottom right, and Nigerians are on the bottom left. And what's really apparent is if I map the self-reported race from our population, that, and this is well known, that the African Americans have a lot of variation along the axis in terms of their proportion of African ancestry. So while this, the idea of race may be separate from this, it still is very interesting to note that genetic differences may absolutely play a part within the African American race category. Furthermore, if we look at the Hispanic Latino, we notice that there's a split along both sides of the PC space, which represented differences, strong genetic differences between Puerto Ricans, Mexicans, and individuals from Colombia. So in our next steps with more data, we've actually already in the process of doing disease networks with more resolution. So with that, I want to thank my advisors, Joel Dudley and Rong Chen, as well as individuals in the Shat Lab at Mount Sinai and the Battelle Lab at Harvard. And thanks again to Akamai Technologies for getting me here. And I'll be happy to take questions now or talk afterwards. Thank you.